Hello, I'm James Graham, CEO of Recce Pharmaceuticals Limited, ASX RCE. Thank you for joining today's Key Opinion Leader event, Clinical Data Milestones and the Recce Innovation Journey. Joining us today is Professor Eugene Athan, Principal Investigator of Recce's recently successful and completed Phase 2 clinical study. Professor Athan will be discussing the study and some of the groundbreaking insights of Recce 327 on a broad range of bacterial infections. Dr. Alan Dunton will be discussing the positive implications of completing this study and the Phase 3 trials just ahead of us. Dr. Sahini Saka, Principal Investigator of a anti-infective research unit at Murdoch Children's Research Institute on Recce's technologies. We hope you enjoy today's presentation.
Hi everyone, good afternoon. My name's Eugene Athan. I'm a Professor of Infectious Disease and I've been the Principal Investigator for an exciting new Phase 2 trial, the Recce product of 327, which is a topical gel which seems to have worked very effectively in the setting of acute bacterial skin and skin structure infections, and I'll be talking a bit about that trial. By way of background, I'm a professor at the School of Medicine. I'm a co-director for a research centre which looks at infectious disease and immunology research at Deakin University, and also I'm a specialist at Barwon Health, one of the largest regional health services in Australia, and I also head up the public health unit at the Barwon Southwest region under the Victorian government. So what is an acute bacterial skin and skin structure infection or diabetic infection? These are common. We see skin and soft tissue infection every day in both general practice primary care and in hospitals. They relate to underlying risk factors, sometimes associated with trauma. In the area of diabetes, it's particularly quite a serious condition for a whole lot of reasons. People lose feeling in their feet, then they can develop trauma, ulcers, complicated infection, and can even result in potential amputation. This is one of the most important causes of disease in diabetes seen globally. And the pathogens, we know golden staph and a whole lot of other potential gram-negatives or superbugs, and in the majority of cases up to 80% are polymicrobial, so different germs, but superbugs relate to antibiotic resistance. The readmission rate for diabetic foot infections is about 40%, and there's a significant mortality at 1 year associated with that. So this is a serious disease and needs major alternative attention besides conventional approaches to treatment. We've seen increasing rates of golden staph or MRSA, methicillin-resistant Staph aureus, patients in hospital, and this is a challenging infection to treat with limited antibiotic options at the moment. So current therapy includes wound care, surgery, as I mentioned, broad-spectrum antibiotics. Acute bacterial skin and skin structure infections are a significant healthcare problem, encompassing indications resulting in necrotizing fasciitis or flesh-eating bacteria, post-operative wound infections, and I've already mentioned diabetic foot infections. There's been no placebo-controlled trials because it is considered to be unethical to withhold standard treatment for patients with infection. What about the Phase 2 Recce 327 topical gel? So this is a product that seems to have extremely good efficacy against many bacteria, so very broad spectrum efficacy. And this is the first time that we've trialled it as a gel to be delivered to skin and skin structure infections or for diabetic ulcers with infection. So the Phase 2 clinical trial is an open label efficacy study, exploratory evaluation of the bioavailability of single and multi-dose R327 topical gel applied to acute bacterial skin structure infection. It's been used in both men and women, only adults, no age limit, upper age limit. The study aimed to provide critical data on the gel's effectiveness in treating a broad spectrum group of acute bacterial skin and skin structure infection, including diabetic foot infections particularly. The trial site at Barwon Health is one of the largest regional health services in Australia, and we provide access to diverse patient populations. What were the endpoints that we achieved during the trial? We achieved all primary and secondary endpoints as an open label trial, evaluating the efficacy, safety and tolerability of the drug. We also looked at pharmacokinetics. So what were the blood levels of the drug, the agent, when it was applied directly to infected wounds? We enrolled 30 patients, 29 included in the final analysis. One patient was withdrawn due to pre-existing pain at the wound site that was not related to the topical treatment. After 7 days of treatment, 86% of patients, so 25 out of 29, treated with our 327 gel had successful clinical response. At 14 days, up to 93% achieved primary efficacy endpoint. In other words, responding in terms of local inflammation or signs of infection. So we can say that R327 gel was demonstrated to be safe and well tolerated, achieving all the study endpoints with no serious adverse events reported. In particular, the study endpoints that we looked at were the Lipsky scale, which measures inflammation in diabetic ulcers or wounds, and we used wound assessment tools as well. And these are validated tools. The FDA accepted diagnosis tools, assessment severity of patient wounds. So the endpoints that I've mentioned to evaluate the efficacy of 327 topical gel in skin and skin structure infection, evaluate the potential for systemic bioavailability of single and multiple dose topical gel applied once daily for 7 or 14 days. The diabetic foot infection, we mentioned the Lipsky scale, clinical resolution of infection, and this is accepted by the FDA as a validated measure of outcome measure. 7 parameters were graded 0 to 3 for 21 total points. The higher the score, the worse the diabetic foot infection. We look for things like erythema or redness, exudate, such as purulent discharge, induration, thickening, tenderness, pain, and warmth. And then the Bates-Jensen wound assessment tool, also accepted by the FDA, shows parameters graded 0 to 5 for 65 points. Higher the score, worse the skin structure wound. We measure things like size, depth, the edge, any undermining of the tissue, any dead or necrotic tissue, any exudate, fluid around the wound, skin color, surrounding changes, swelling or edema, thickening or induration, granulation of healing, and epithelialization. So these are the methods that we use to evaluate patient wound healing, and subsequently rated patients as either cured or improved, and they're validated and accepted by the FDA. So the potential for Recce 327 gel for future applications. We saw no serious adverse events during our trial. The results demonstrated the potential to address the skin and skin structure infection market. And this has been a considerable challenge, as I've already said, big burden of disease to the healthcare system. And those at high risk of skin infections and poor outcomes are often diabetic patients in particular. We saw a rapid onset of efficacy of 327. People responded mainly within the first week. And there's obviously a much more significant clinical setting for the potential of this agent to be used in different infection contexts. So plenty of therapeutic potential. And these are my references. So thank you for your attention.